This video is going to teach you how to correctly complete a Punnett square to answer questions about genetics. There are 5 steps to this process, and every step is required, just like problems in math class. The first step is to assign genetic symbols. Identify the dominant allele and its symbol. In our example today, we're going to be talking about height of pea plants, just like Gregor Mendel did with his famous experiment. The dominant allele in that case was the allele for tall plants, and the symbol for this dominant allele will be a capital T. Next, list the recessive allele and its symbol. So short was the recessive allele, and its symbol is going to be a lowercase t. The second step to completing Punnett squares is to write out the genotypes of the parents that will be crossed. So you need to write out the father's genotype, and our father plant is going to be capital T, so homozygous dominant, and the female mother plant is going to be homozygous recessive, lowercase. Remember that this is an example, and the parents' genotypes can be different for every situation, and this is why you need to write out the parents' genotype each and every time. The third step is to set up the Punnett square. So let's just remind ourselves, this is the father's genotype, homozygous dominant, mother's genotype, homozygous recessive. What we need to do is take the father's genotype, which I'm going to be actually now showing in blue, and for each of his alleles, you divide them and put them on top of each column. So the father's alleles, capital T, get written above columns. The mother's alleles get written down the left side, so that her first allele is lowercase t. Her second allele is still, lowercase t. Cross that off there. Okay, now that we have our Punnett square set up, let's move on to the fourth step. The fourth step is to fill in the Punnett square. You do this by copying the father's alleles straight down the column. So this father's alleles first moves here, capital T, and then it gets copied down there for capital T. Same with his second allele, copied here, and capital T. To fill it in for the mother's alleles, her alleles gets copied sideways. So this red allele goes right to this box, and again right to this box. So this is our completed Punnett square. Yes, it looks messy, but I wouldn't recommend drawing the arrows each and every time. So when we clean this up, it will look like this, and our fifth step to completing Punnett squares is to list the genotypes, phenotypes, and ratios. Every box in the Punnett square tells you a possible genetic combination an organism may inherit from its parents. So copy each genotype that was formed in the boxes off to the side or below. In our case, we only have a genotype, one genotype. This is the heterozygous, capital lowercase. Then, once you've listed every genotype, you determine what the phenotype would be. So if you remember that the capital T was the dominant, well, now it looks like an I, but the capital T is dominant, and that is the allele that codes for tall, this plant will be tall. The last thing you need to do is figure out the ratio of genotypes and phenotypes. The capital T, lowercase t ratio is figured out by counting the number of boxes that it appears in. In this case, it appears in 4 out of 4 boxes, giving us a ratio of, well, a percentage of 100%. The phenotype, likewise, is also going to be 4 out of 4. So the tall phenotype will also equal 100%. This should match Mendel's famous experiment where the F1 generation, after crossing a purebred tall and a purebred short, the F1 generation had every single individual as tall. So this is a quick example of how to complete a punnett square.